Assalamualaikum. Dear student, in today's lecture, we will discuss about meiosis. As you know, the meiosis is a reduction division, and in which the number of chromosomes reduced to half. And meiosis contain two divisions. The first one is meiosis 1, and the second one is meiosis 2. First thing that we will discuss what is meiosis. As I already discussed, the meiosis is a cell division by which the gamete with the half number of chromosomes are produced. And because all the cells of the body are diploid except gametes, and gametes are two. The first one is male gamete, and the second one is female gamete. And meiosis is always occur during sexual reproduction. Fertilization, as you know, fertilization is actually the fusion of sperm and fusion of egg, and they form a zygote. When they form a zygote, then multiple divisions taken place in zygote that form in embryo, and after embryonic phase, different gastrula stages, yeah, gastrulations occurs, in which ectoderm, endoderm, and mesoderms are formed, and from ectoderm, different body parts formed, and from mesoderm and endoderm, actually these are the germ layers that form all the body parts of the organisms. So meiosis yeah, occurs during sexual reproduction, and in which the gametes are required. And there, as you know, there are two gametes, the sperm and egg. Gametes have half number of chromosomes, and as you know, the egg is formed in ovaries, and the sperm form in testes. And testes and ovaries both are called gonads, and the gonads are reproductive structures in organisms. And the formation of sperm is called spermatogenesis, and the formation of egg is called oogenesis. And meiosis is similar to mitosis with some chromosomal differences. First, we will discuss what is spermatogenesis, and the second, we will discuss about oogenesis. As you know, the genesis means formation, and spermato means sperm, which means the formation of sperm is called spermatogenesis. Here is a human sex cell which is diploid, and after meiosis 1, the one cell divided into two halves, each contain haploid number of chromosomes. And then meiosis 2 will be takes place, and from each cell, two further cells will be produced, and from a single parent cell, four haploid cells are produced, and with a half number of chromosomes. As you know, as you can see here, there are 46 chromosomes, and the daughter cells contain 23 chromosomes. It means a reduction deviance takes place here. And on the screen, you can see the meiosis of mouse testes, and here is a parent cell, and here is a first division, here is a second division, and as a result, four gametes formed. Then interface, as you know, before division, the cell prepared itself for division. So when a cell divide, it have two phases. The first one is interphase, and the second one is M phase. M phase might be mitosis or meiosis, and in interphase it is a preparatory phase, and the cell contain 90% interface, and a cell spend most of the time in interface. And as you know, the interface contain different phases: the G1 phase, S phase, and G2 phase. And in G1 phase, all the things are prepared which need in S phase. And in S phase, S means synthesis, and duplication or replication of the DNA takes place. So each cell contain equal number of chromosomes. So in interphase, S phase will be done, and each duplicated chromosome that consists of two identical sister chromatid will be attached at the same centromere. And centrioles also replicate, and centrioles are actually a non-membrane bounded organelle which is present at the opposite pole of the cell, and they form spindle fibers that help to pull the chromatids or chromosomes towards opposite poles, so the cell divide into two cells. So Nucleus and nucleolus in interface is visible. As you can see, here is a chromatin, and here is a cell membrane, here is a nuclear membrane, and here is a nucleolus. Nucleolus is a dark portion of the nucleus, and chromatin is a thread-like structures which is present before cell division, and after that the chromatin condense and form a chromosomes, and after interphase there are different phases takes place. So meiosis 1 contain 4 phases. The first one is prophase 1 because it is a division of meiosis 1. So prophase 1, then metaphase 1, anaphase 1, and telophase 1. So in prophase 1, it is the longest and most complex phase, contain about 90% of the cell cycle, and 90% of the prophase 1. And in this phase, chromosomes condense, synapse occur. It means the homologous chromosomes come together to form a tetrad. Always keep in mind, meiosis always takes place in homologous chromosome. Meiosis never takes place in chromosome. Homologous chromosomes are actually the chromosomes which are same in size and shape. So tetrad is 2 chromosomes at 4 chromatids. As you can easily see here, here is one chromosome, and this is one chromosome that contain 2 chromatid, and both these chromosomes are homologous chromosomes, and here is a pair of homologous chromosomes. Um, this is one chromosome and this is second chromosome. So here is a centromere that attach two sister chromatids with each other. So for example, just suppose here is a male chromosome and this is a female chromosome, and they will form a synapse. They come close to each other and they will do crossing over, so the chromosome exchange so genes can be exchanged in next generation. So here is a homologous chromosomes, here is a sister chromatid, and here is a tetrad. Homologous chromosomes is actually the pair of chromosomes which is maternal and paternal that are similar in shape and size. Homologous chromosomes pair, and this pairing is called tetrad, that carry the genes controlling the same inherited trait. And what is locus? Locus is actually the position of a gene on a chromosome. So humans have 23 pair of homologous chromosomes and total 46 chromosome in one cell. So 22 are autosome chromosome out of 23 pairs, and the first one pair is a sex chromosome. The first 22 pairs are autosome, which are same both in male and female, and the 23rd pair is different. It will be XX in female and it will be XY in male. So karyotyping is actually the method in which the chromosome of a cell in relation to number, size, and type are studied. And here are different 46 chromosomes which are present in one cell of human or organism. And all the chromosomes are different on the basis of their centromere and on the basis of their shape. Homologous chromosomes, for example, here is a eye color gene and here is a hair color gene, and in second parent here is a eye color gene and here is a hair color gene, because we are talking about homologous chromosome, and homologous chromosome that contain the same genes and same shape and same size of the chromosome. So the one is paternal and the second one is maternal. So here you can see the 46 chromosome karyotyping of 23 pair of homologous chromosomes are 46 chromosomes, and all the chromosomes are different on the basis of their shape of centromere. 22 chromosomes are autosome chromosome, and 23rd chromosome is a sex chromosome. Can you see the extra 23rd chromosome here? As you can see here, all the chromosomes should be in pair, but when a chromosome carry extra copy, this is not normal. It could be a disease. So here, if any chromosome contain 3 copies, as you can see here is a 23rd chromosome contain 3 copies, it means this disorder is a Down syndrome, and Down syndrome is not a normal. Down syndrome person has a broad jaw, protruded eyes, protruded tongue, and mentally retarded person. So it could be done due to non-disjunction of the chromosome and due to unequal distribution of chromosome between two cells. In humans, the sex chromosomes are the 23rd sex and 23rd set, and it could be XX in female and XY in male. And XX chromosome both are same, but Y chromosome is smaller than X chromosome. So here you can distinguish the original picture of X chromosome and Y chromosome. Y chromosome is smaller than X chromosome. So as we're talking about prophase 1 and we discussed about synapse formation, tetrad formation, the next step is crossing over. Crossing over means variation may occur between non-sister chromatid at the chiasmata formation. Chromatid, as you know, the one chromosome contain two chromatid, and crossing over always takes place between non-sister chromatid. Crossing over, segment of non-sister chromatid break and reattach to the other chromatid. Chiasmata are the sites of crossing over. Chiasmata formation is actually the site of crossing over, and you can see easily when the two chromosomes, when the two homologous chromosome join in such a way that a X-shaped structure formed, and that X-shaped structure actually shows the chiasmata formation. Yeah, crossing over non-sister chromatid. Here you can see both chromatid are sister chromatid, and both these are also sister chromatid, but this and this chromatid both in relation to each other are non-sister chromatid, and both these non-sister chromatid will exchange their genes with each other. And so this X-shaped structure is called chiasmata formation, and after crossing over, that chromatid contain the part of this chromatid, and this chromatid contain the part of that chromatid, and this is the result of variation, and that's why all the individuals, even the family members, are different from each other due to this crossing over. Then the next one is sex chromosomes. As we already discussed, both XX are same and XY are different. XX chromosomes are female chromosome and XY are male chromosomes. Uh, Now we need to discuss prophase 1, but we already discussed about crossing over. So here a cell in which the nuclear envelope disappears, spindle fibers are formed, ester fibers are formed, and centrioles actually form the spindle fibers, and the homologous chromosomes are present inside the cell. Then metaphase 1, meta means equator, and this is the shortest phase because in homologous chromosomes will align in the center of the cell. So independent assortment occurs here. Independent assortment actually described by Mendel, and Mendel was a father of the genetics. And here you can see the orientation of homologous chromosomes to poles is random, and the variations, and here you can learn easily. For example, 2n means 4, then n is equal to 2, and it means the 4 combinations could be takes place. Metaphase 1, and the cell visible like this because the cells are aligned at the equator of the cell. Here is a question in terms of independent assortment: how many different combination of sperm could a human male produce? So the answer will be according to formula 2n. 2n is equal to 23. It means n is 23. So 8 million combinations a human can produce with different gametes. So the next one is anaphase. In anaphase, the chromosomes start to move towards opposite pole, and the whole chromosome move toward the opposite pole. Here is not here we're not discussing about the mitosis where the chromatid separated and chromatid moved to opposite pole. Here we are discussing about the meiosis. So the whole chromosome and whole two sister chromatid that are attached at their centromere, they will start to move, they will start moving towards opposite pole. In anaphase, as you can see here clearly, and here is crossing over takes place. So that chromatid contain different chromatid. In telophase 1, each pole now has a haplite set of chromosome, and before that karyokinesis takes place, and after karyokinesis cytokinesis takes place. And now the cyto means cytoplasm and kinesis means division. It means that cytoplasm will be divided, and this is called cytokinesis, and before that the nucleus divides, so it is called karyokinesis. So in telophase 1, before cytokinesis, two cells look like this, and after that, in plant and in animal, the cytokinesis is different because plant contains cell wall. So the phragmoplast is formed at the center of the cell, which is formed by the Golgi apparatus. But in animals, the cytokinesis takes place with the help of cleavage or with the help of protein, actin, and myosin that divide the cell into different cells. So as you can easily see, here are whole chromosomes separated. Here is not the chromatid that gets separated. So these both cells need to divide once more. So when they divide again, this division will be division 2. So we can call this division is a meiosis 2. So meiosis 2 is quite similar. There is no interphase, there is no synthesis. There is no synthesis of the DNA takes place here. A very short, no more DNA replication. And meiosis 2 is similar to mitosis because in mitosis the chromatid gets separated, and in meiosis 2 also the chromatid gets separated. And this question is very important. So prophase 2, it also contain all the phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Same as prophase in meiosis. So in prophase, the nuclear envelope disappear, and centrioles form spindle fibers that are attached at the kinetochore protein, which is present around the centromere. So in metaphase 2, the chromosomes are aligned at the equator, and in anaphase 2, the chromatid start moving towards the opposite pole. And in telophase 2, the chromosomes nuclei again form, cytokinesis occur, 4 haploid daughter cell formed, and each cell have haploid are half number of chromosomes. So gamete could be sperm or egg. So telophase 2, here you can see the gamete gets separated and the 4 cells are formed, and these 4 cells have half set of chromosomes. It means reduction division takes place. I hope today's lecture is helpful for you. If you like the video, please like, share, comment, subscribe the channel, stay connected. Allah